Today we will be discussing our research on anthocyanins in Z. maize L, otherwise known as purple corn, as inhibitors of Escherichia coli.
Naturally occurring bacteria in the gut is beneficial in maintaining one's overall health. Anthocyanins, which are pigments found in fruits and vegetables, are theorized to regulate gut bacteria which promote gut function. This study focuses on anthocyanins in Z. maize L and their potential ability to support commensalism while inhibiting enteroinvasive E. coli.
As mentioned, anthocyanins are molecules that contribute to the pigment in many foods, especially berries, as well as other fruits and vegetables. The figure on the right shows the basic structure for all anthocyanins, with the pink R's representing the different variations of chemical groups that can be added onto this basic structure and differentiate each anthocyanin. The figure on the bottom left shows the range of these pigments produced by the anthocyanins. Foods containing anthocyanins typically have a combination of these that produce a very unique pigment in each food.
Now, you might be asking, why study anthocyanins? First, bacteria has evolved resistance to antibiotics, meaning that other substitutes might need to be used against bacteria. The answer could be found within anthocyanins, which have shown to have health benefits such as antimicrobial and antioxidant properties. After extensive research, purple corn was chosen because of the variety of anthocyanins and health benefits. According to Layout et al., purple corn had potent antioxidant, anti-inflammatory, anti-mutagenic, anti-carcinogenic, and anti-geogenesis properties. We first had to obtain crude extract of purple corn before we could test the anthocyanins on E. coli. Purple corn powder was mixed with acidified ethanol, and 2 samples were vortexed and 3 samples were sonicated. We then centrifuged, decanted, and filtered 1 milliliter of these samples in order to conduct a high-performance liquid chromatography, also known as HPLC. The HPLC was crucial to identifying individual anthocyanins within purple corn. On the right side of the chart, you can see the measurements we used for the analytical HPLC.
The image here shows the results of HPLC using the mixed samples of crude extract of Z. maize L. Looking at the graph on the lower left, peaks indicate the presence of isolated anthocyanin, with the x-axis showing retention time in minutes. If you take a look at the purple arrow at around 7.5 minutes of retention time, this is a common place where we would see the anthocyanin pelargonidin 3-glucoside. And if you look at the orange arrow at around 9 minutes of retention time, this is where we would commonly see the anthocyanin peonidin 3-glucoside. This image shows that in our sample, we were able to isolate these 2 anthocyanins. This also tells us that these anthocyanins will most likely be providing the benefits in purple corn. Similarly, the image here shows the results from an HPLC, this time with sonicated samples. Again, the graph in the lower left corner shows peaks where the isolated anthocyanins would be. Looking at the purple arrow at around 6.5 minutes of retention time, this is commonly when we would see the anthocyanin cyanidin 3-glucoside. The pink arrow at around 7.5 minutes of retention time is where we would typically see the anthocyanin pelargonidin 3-glucoside. Lastly, at around 9 minutes of retention time, shown by the orange arrow, is where we would typically see the anthocyanin peonidin 3-glucoside.
Commensal bacteria, which are frequently referred to as good bacteria, are the normal and naturally occurring microflora within the body. Commensal microbiota are essential to human health. They aid in digestion as well as help in the development and optimal functioning of the host's immune system. Commensal bacteria act on the host's immune system to induce protective reactions that prevent colonization and infection by pathogens by producing antimicrobial substances. Although some bacteria are healthy living in the body, not all are, which is why it is important to see if foods that include anthocyanins can help promote good bacteria while inhibiting pathogenic bacteria. The commensal bacteria we tested were ATCC 8729, ATCC 25922, and ATCC 43895.
With the commensal strains of E. coli we conducted at MIC, we first added 1 to 5 milligrams of freeze-dried purple corn to dimethyl sulfoxide in order to produce a 55 and 0.5 milligram per milliliter solution that will be used later in step 4. Then, a 1 to 10 dilution of inoculated bacteria to LB media was created in order to discover the optical density of each bacteria. After creating the correct bacteria to LB solution, we tested the mixture against several of our anthocyanin concentrations via MIC. Our results show that the E. coli was resistant to the anthocyanins at all concentrations.
All of the enteroinvasive bacteria we tested were strains of E. coli. Bad bacteria are strains that result in disease and illness. The most common strains of E. coli that make you sick do so by producing a toxin. This toxin damages the lining of your small intestine and normally causes diarrhea. Most cases of E. coli infections are mild. However, some strains can cause severe symptoms and even life-threatening complications, which can lead to kidney failure and death. These complications that are caused by enteroinvasive bacteria are one of the reasons why we wanted to test a natural way to inhibit its growth. The enteroinvasive bacteria strains we used were NR712, BAA 1883, and BAA 3236.
After testing Z-Maze-L against commensal strains of E. coli, we moved on to conduct an MIC with the enteroinvasive E. coli strains. Following a similar procedure as before, we obtained the optical density for each bacteria strain by creating a 1 to 10 dilution with LB media. Then, using our calculated optical density, we tested the inoculated media against various concentrations of previously measured out freeze-dried purple corn with dimethyl sulfoxide. Once again, the results showed that the bacteria strains were resistant at all concentrations. This shows that the anthocyanins in purple corn do not have enough antibacterial property to affect our E. coli strains.
After conducting the MIC, we discovered that the commensal and enteroinvasive E. coli was resistant against all anthocyanin concentrations. Future directions will be to test stronger concentrations to discover if enteroinvasive E. coli will be susceptible and commensal will remain resistant. We will also attempt to isolate individual anthocyanins via the HPLC to identify if the enteroinvasive E. coli is susceptible to one specific anthocyanin.
Thank you for taking time to listen to our research presentation. We would like to say thank you to Dr. Blackledge, Dr. Womack, and Dr. Hughes for working with us. We'd also like to thank the High Point University Biology and Chemistry departments, as well as the Natural Science Fellows, for providing resources for our study. Lastly, we'd like to thank HyPerks for having us. Thank you.